Good day to the viewers. Today I am going to discuss about qualitative analysis of carbohydrates. First, let's see about the introduction of carbohydrates. Carbohydrates are defined as the polyhydroxy aldehydes or polyhydroxy ketones. Carbohydrates are the key source of energy used by living things. It is widely distributed in plants and animals. It also serves as extracellular structural elements are present in cell wall of bacteria and plants. They are the primary fuel source for the brain and muscles during physical activity. Carbohydrates are broken down into glucose, which is either used immediately for energy or stored in the liver and muscles as glycogen for later use. The general molecular formula of carbohydrate is CnH2nON, which means the N number of carbon, hydrogen, and oxygen. Carbohydrates are classified into simple and complex sugars. Simple sugars are monosaccharides which contain single sugar molecules and which cannot be further hydrolyzed. Examples of monosaccharides are glucose, fructose, galactose, and mannose. Complex sugars consist of more than one sugar molecules. Based on the number of sugar molecules, complex sugars are classified into disaccharides, oligosaccharides, and polysaccharides. Disaccharides, it consists of two sugar molecules. Examples are maltose, lactose, and sucrose. Maltose consists of two molecules of glucose. Lactose consists of one molecule of glucose and one molecule of galactose. And sucrose contains one molecule of glucose and one molecule of fructose. Oligosaccharides, which contains 3 to 9 monosaccharide units. Examples are raffinose and galacto-oligosaccharides. Raffinose is composed of glucose, galactose, and fructose. Galacto-oligosaccharides typically consist of 2 to 8 galactose units linked to a terminal glucose unit. Polysaccharides are complex carbohydrates which is composed of more than 9 monosaccharide units. Examples are starch, glycogen, inulin, cellulose, etc. And these complex carbohydrates can be broken down into simple sugars units through a process known as hydrolysis. When discussing carbohydrates or other biological substances, qualitative analysis and quantitative analysis are two fundamental approaches used to describe and measure their characteristics. Qualitative analysis involves identifying the presence or absence of a particular substance or component without measuring its exact amount, whereas in quantitative analysis involves measuring the exact amount of concentration of a substance in a given sample. Now let's see about the various qualitative tests routinely used to identify the presence or absence of carbohydrates in a given test solution. First, Molish test. This is a general test for the identification of carbohydrates. In the presence of sulfuric acid, the sugar undergoes dehydration to furfural or hydroxy for methyl furfural, which condenses with alpha naphthol, resulting in the formation of a violet or purple colored ring. Here procedure is: in 2 ml of test solution, add 2 drops of Molish reagent and gently mix the contents. Then carefully add 1 ml of concentrated sulfuric acid along the sides of the test tube. Do not shake the test tube while adding the acid. A violet or purple ring appears at the interface of sugar solution and the acid. It indicates the presence of carbohydrate. Iodine test is used to identify the polysaccharides. Principle: molecular iodine reacts with the polysaccharides, forming a colored adsorption complex. Variations in the color of the polysaccharide iodine complex is observed as starch gives blue color while glycogen forms a red-brown color. Procedure for iodine test is: add 1 ml of test solution with few drops of iodine solution, then the appearance of blue color which confirms the presence of starch. Next, move on to Benedict test. It is used to detect the presence of reducing sugars such as glucose, fructose, maltose, etc. Here the principle behind the Benedict test is reducing sugars have free aldehyde or free ketone groups capable of acting as a reducing agents. When these sugars are heated in an alkaline solution, they can reduce metal ions. When Benedict reagent is mixed with a solution containing reducing sugars and heated, the reducing sugars reduce the cupric ions to cuprous ions. The cuprous ions then precipitate as insoluble cuprous oxide, which has a distinctive color change depending on the concentration of reducing sugar. Procedure: 0.5 ml of test solution is mixed with 2 ml of Bendick reagent and keep the test tubes in a boiling water bath for a few minutes. Formation of red precipitate is obtained. Here, the blue color indicates that no reducing sugars are present. Green color represents the low concentration of reducing sugar. Orange color indicates high concentration of reducing sugar, and the red color denotes very high concentration of reducing sugar. BAFO test is performed to distinguish between reducing monosaccharides and disaccharides based on their ability to reduce cupric ions under acidic conditions. Here the principle for BAFO test is reducing sugars are oxidized by the copper ion in solution to form carboxylic acid and a reddish precipitate of cuprous oxide within 3 minutes, where reducing disaccharides undergo the same reaction but do so at a slower rate of 7 to 12 minutes. Procedure. Here 1 ml of test solution is mixed with 2 ml of BAFO reagent. Then keep the test tube in a boiling water bath for 3 minutes. The formation of red precipitate reveals that presence of monosaccharides. Next, Bial's test is used to distinguish between the pentose and hexose sugars. When pentoses are heated with Bial's reagent, they undergo a dehydration reaction in the presence of hydrochloric acid. The arsenol reacts with the dehydration products of the pentoses to produce a green or bluish green colored complex. However, presence of hexose sugar produces a brown color complex. Procedure. Add 0.5 ml of test solution with 2 ml of Bial's reagent and keep it in boiling water bath for 5 to 10 minutes. Appearance of bluish green exhibits the pentose sugars. Next, Selivanov test. This test is used to differentiate between the aldose and keto sugar. Principle is keto sugar such as fructose when exposed to acid medium undergo dehydration forming hydroxymethyl furfural more rapidly than aldohexose. This furfural condenses with resorcinol to produce cherry red color. Procedure. 1 ml of test solution is mixed with 2 ml of Selivanov reagent. Then keep the test tube in boiling water bath for 5 minutes. The formation of cherry red color denotes the presence of keto sugar. The last one is ozone test. It is a confirmatory test. Principle. This test is for reducing sugars with free aldehyde or ketone group forming ozone crystals. And non-reducing sugars like sucrose do not form an ozone. When phenylhydrazine reacts with reducing sugars at a boiling temperature, ozone is formed. Phenylhydrazine reacts with carbonyl compounds in neutral or slightly acidic conditions to give phenylhydrazone, which is highly soluble. When hydrazone reacts with further phenylhydrazine molecules, the condensation products formed are insoluble, which precipitate out as crystal. Procedure. 3 ml of test solution is mixed with 0.5 gram of phenylhydrazine and add 0.1 gram of sodium acetate. Then add few drops of acetic acid. Then mix well the contents and keep the tubes in a boiling water bath. Once the yellow precipitate is obtained, allow the test tube to cool down to room temperature. The shape of the crystal will be seen under a light microscope. For fructose, the yellow precipitate will be obtained within 2 minutes and the needle shape can be viewed under the microscope. For glucose, it is about 5 minutes and the crystalline structure of glucose is needle or broomstick shape. For galactose, it is about 20 minutes and it forms thorny ball or rhombic plate shape. For maltose, it is about 30 to 45 minutes. It forms sunflower or petal shaped crystal structure. And for lactose, it is about 30 to 45 minutes. The crystalline structure of lactose is puff shaped or cotton balls. Thank you for listening the video.